Hello, friend. Welcome to Science for Sleep. There is something about ants that seems almost unbelievable. An animal only a few millimeters long can lift, drag, or carry several times its own body weight. That strength is impressive on its own, but it becomes even more interesting when we notice how ants use it in the ordinary places around us. An ant crossing a kitchen floor, an ant moving over a garden stone, an ant pausing beside a narrow crack in the pavement. At first, she may seem to be wandering. She stops, turns, and sweeps her two elbowed antennae across the ground. Then she walks a few body lengths, changes direction, and continues toward a place that looks empty to us. But she is probably not moving at random. Her antennae are testing the surface for chemicals left by other ants. Her feet are feeling the texture beneath her. Her body is also responding to warmth, moisture, light, and the ants she has recently met. The ant before us is almost certainly female. If you enjoy these quiet journeys, you're welcome to subscribe. And if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find this calm corner of science, one sleepy listener at a time. In most ant species, nearly every worker outside the nest is a daughter of the colony's queen. She may search for food, carry soil, defend an entrance, clean another worker, or help feed the young. The worker continues across the surface, but no ant inside the nest has sent her a detailed set of instructions. The queen has not told her where to walk. Despite the familiar name, an ant queen is not a ruler in the human sense. Her main role is reproduction. She produces the eggs from which new workers, males, and future queens may develop. She does not study a map of the nest, count the food stores, or assign jobs to thousands of daughters. The worker does not possess that full picture either. She knows only what reaches her through direct contact. A scent trail may make one route easier to follow. A hungry nestmate may accept more liquid food from her. A damaged tunnel may release loose soil and bring nearby workers toward the same place. An alarm chemical may cause her to move faster, open her jaws, or recruit others. Each response is small. Yet when hundreds or thousands of workers respond to one another, organized patterns begin to appear. A food trail grows stronger. Brood is moved to a warmer chamber. An entrance is repaired. A threat is surrounded. No individual ant needs to understand the complete result. This kind of system is called decentralized organization. Decentralized means that control is spread across many individuals instead of coming from one leader. Some ant colonies contain only a few dozen workers. Others contain hundreds of thousands. Certain army ant and invasive ant societies can reach into the millions, with connected nests spread over great distances. When scientists estimated the number of ants living on Earth in 2022, they arrived at about 20 quadrillion. That is 20 followed by 15 zeros. At the time of the estimate, it worked out to roughly 2.5 million ants for every human being. Their combined dry carbon mass was estimated at around 12 million metric tons. Dry carbon is a way of comparing living material after removing water and measuring the carbon held inside it. Those enormous totals are built from individuals small enough to disappear beneath a leaf edge. The worker we began with may be only a few millimeters long. She may carry a crumb several times her own weight, follow a fading trail, and enter a gap barely wider than her head. Once she passes below the surface, she becomes part of a flow of workers moving food inward, waste outward, and information in both directions. Her colony may have no commander and no single body. Still, it can gather food, raise young, choose a new home, repair damage, and survive for many years. To understand how such a system became possible, we have to look far beyond the worker's brief journey across the floor. Her story reaches back more than 100 million years, to small wasp-like ancestors living while dinosaurs still walked the earth. Long before a worker followed a scent trail into the ground, her lineage belonged to a wider family of insects. Ants are members of an order called Hymenoptera. That group also includes bees, sawflies, and wasps. The name refers to the thin, clear wings found on many of its members, although worker ants usually have no wings at all. Termites belong elsewhere on the insect family tree. Their closest living relatives are cockroaches, and their social colonies developed along a separate path. An ant colony may resemble a termite colony from a distance, with queens, workers, tunnels, and shared care of young. The resemblance comes from similar problems producing some similar solutions. Ants began from a wasp-like starting point. Their early relatives already possessed several useful features. Strong mandibles could cut, grip, and carry. A narrow waist allowed the rear of the body to bend with greater freedom. Venom glands and stinging structures provided defense or helped subdue prey. Females laid eggs, built or used nests, and cared for developing young. Evolution worked by altering these existing parts and behaviors over many generations. Small changes in anatomy, nesting, food gathering, and brood care could become more useful when related females remained together. Cooperation could then grow from behaviors that had once served the mother and her own offspring. The earliest clearly recognizable ants appear in rocks and amber from the Cretaceous period. This long span of Earth history lasted from about 145 million to 66 million years ago. Dinosaurs still occupied the land, while flowering plants were spreading into more forests and open habitats. One famous early ant, Sphecomerma freyi, was found in amber from New Jersey. The amber is about 92 million years old. Its body carried a mixture of features. The narrow waist and elbowed antennae pointed toward modern ants. Other parts, including the shape of its head and mandibles, still resembled its wasp-like relatives. The fossil was described in 1967 after 2 pieces of amber were collected near the town of Cliffwood. A tiny insect sealed inside hardened tree resin helped confirm that ants were already present, while the Cretaceous forests were growing around dinosaurs. Other fossils reveal branches that left no modern descendants. Among the strangest are the extinct insects, often called hell ants. Several species have been preserved in amber from roughly 100 million years ago. Modern ant mandibles usually close from the sides, meeting in front of the mouth. Hell ant jaws moved upward in a vertical direction. Some species also had horn-like structures on the head. A prey animal may have been lifted between the rising jaws and the projection above them, creating a trap unlike anything used by living ants. This unusual system lasted for tens of millions of years before disappearing near the end of the Cretaceous. Its loss matters because it shows how many ant designs once existed. The bodies seen today belong to the branches that continued, while other successful forms vanished with changing habitats and mass extinction. Ant diversity grew as land ecosystems became more complex. Flowering plants produce seeds, nectar, fruit, wood, and fallen leaves. Plant-feeding insects became new sources of prey and sugary liquid. Leaf litter created cool, damp layers filled with small animals, fungi, and decaying material. Trees offered bark cracks, hollow twigs, roots, and spaces high above the ground. These changes opened many ways to find food and build nests. Some lineages became hunters beneath leaves. Others moved into wood, soil, deserts, or forest canopies. Social life also developed in different forms, from small colonies with a few workers to societies containing enormous specialized populations. No single moment created the finished ant colony. Anatomy, communication, brood care, and cooperation were shaped together across deep time. That history now rests inside every worker body, in the structures used to touch, smell, lift, cut, sting, and pass information from one ant to another. The long history of ant evolution can still be seen in the shape of a worker moving across the ground today. At the front is the head, carrying the main tools used to sense and handle the world. Behind it is the mesosoma, the firm middle region, where all 6 legs connect. The word mesosoma means middle body. In ants, it includes the true thorax and one abdominal segment that became joined to it during evolution. A narrow waist follows. This waist is called the petiole, and it may contain one small segment or two, depending on the ant group. Beyond it sits the rounded rear portion known as the gaster. Much of the digestive system is held there, along with defensive glands and the reproductive organs of queens. Every part is enclosed by an exoskeleton. An exoskeleton is a supporting framework worn on the outside of the body. Its main material is chitin, a strong biological substance also found in the shells of crabs and the bodies of beetles. The shell protects soft internal tissues and gives the muscles firm surfaces to pull against. A thin, waxy layer helps slow the loss of water, which becomes especially important for ants crossing hot soil or dry leaves. This outer armor cannot expand continuously. Ants grow mainly while they are larvae, shedding an old covering in a process called molting. Once a worker reaches adulthood, her hard exoskeleton fixes most of her final size. The busiest sensory structures rise from the head. Each antenna has a long first segment followed by a clear bend, producing the elbowed shape shared by almost every modern ant. The surface is covered with tiny sensory structures called sensilla. Some respond when the antenna touches a grain of soil or another ant. Others detect moisture, moving air, temperature, and chemical molecules. An antenna sweeping across the floor may gather information from several sources within a few seconds. A faint chemical trace can reveal where another worker walked. Direct contact with a nestmate can provide clues about her colony, age, task, and reproductive condition. Odor matters so much that many ant genomes contain unusually large families of odorant receptor genes. These genes provide instructions for making receptor proteins that respond to particular kinds of airborne or surface chemicals. A worker never receives the colony's entire supply of information. She samples what lies within reach of her antennae, then carries the effects of that contact into later encounters. The jaws below the antennae are called mandibles. Their shapes differ greatly among species. Seed harvesting ants may have broad surfaces suited to crushing. Predatory species can carry long jaws that close around moving prey. General workers use their mandibles to loosen soil, cut food, move eggs, carry larvae, groom nestmates, and grip objects several times wider than their heads. A worker carrying a larva holds the soft body carefully between the inner edges of her jaws. The same structures can close firmly during defense. Her narrow waist adds another kind of movement. Flexible joints around the petiole allow the gaster to bend beneath or around the rest of the body. A stinging ant can curl the tip forward to deliver venom. Some species use the same motion to spray defensive chemicals. Foragers may lower the gaster toward the ground while laying a scent trail from a gland near its tip. The eyes add visual information where light is available. Compound eyes are built from many small units, each receiving part of the scene. Their size varies widely. Ants that work in bright, open habitats often have larger eyes than species spending most of their lives underground. Winged queens and males commonly possess three simpler light-sensitive organs called ocelli on top of the head. These small eyes help detect brightness and assist with orientation during flight. Information also enters through the legs. Sensitive organs detect vibrations passing through soil, wood, leaves, or nest walls. Some ants create vibrations through stridulation, which means rubbing a ridged body surface against another part. The resulting signal can travel through the ground even when airborne sound remains weak. Inside a colony, thousands of workers repeat these small acts of sensing. Antennae touch bodies and walls. Feet detect changes beneath the nest floor. Chemical traces remain for seconds, minutes, or longer. The dark passages are therefore filled with identities, warnings, directions, and requests that human senses cannot directly perceive. A molecule resting on the ground can change what a worker does within a fraction of a second. When that molecule reaches a matching receptor on her antenna, it may encourage her to turn, slow down, follow a path, approach a nestmate, or prepare for danger. Chemicals used this way are often called pheromones. A pheromone is a substance released by one member of a species that changes the behavior or body of another member of the same species. Ant colonies use many kinds of pheromones produced by different glands and released under different conditions. A trail chemical can guide workers toward food. An alarm signal may spread rapidly after a nest is disturbed. Recruitment chemicals bring additional workers toward prey, a damaged entrance, or a site that needs excavation. Sex pheromones help winged males and queens locate suitable mates during reproductive flights. Developing eggs and larvae carry odors that affect how workers clean, feed, sort, and move them. Fertility-related chemicals reveal that a queen is actively reproducing and can influence the reproductive behavior of nearby workers. These signals vary greatly among ant species. One colony may release a trail chemical from a gland near the tip of the gaster. Another species may produce its main trail substance in a gland connected with the venom system. The chemical itself must suit the job it performs. An alarm signal often needs to spread quickly through the air and disappear soon afterward. Chemicals that evaporate easily are described as volatile. Their molecules leave a surface readily and travel through the surrounding air. A short-lived alarm cloud can alert nearby workers without keeping the colony agitated long after the disturbance has passed. Trail pheromones often remain useful for a longer period, although they also fade. Consider a forager returning from a drop of fruit juice. As she walks home, The tip of her gaster touches the ground at intervals and leaves a narrow chemical path. A worker emerging from the nest encounters the trail with her antennae. The concentration is slightly stronger on one side of her body, so she turns toward that side and continues sampling as she moves. If she reaches the fruit juice, fills her crop, and returns successfully, she may add more pheromone to the same route. Each successful journey raises the chance that another worker will follow. The trail can strengthen rapidly when the food is rich and easy to collect. A dry crumb or an empty feeding site produces a different result. Workers returning without food may lay little or no trail chemical. Older pheromone continues evaporating or breaking down on the surface. Traffic then decreases, without any ant formally closing the route. This is a chemical form of feedback. A useful path gains reinforcement through repeated success. A failing path gradually loses its influence as fewer workers renew it. Chemical information also covers the body of every ant. The exoskeleton carries wax-like substances called cuticular hydrocarbons. Cuticular refers to the body's outer covering, while hydrocarbons are molecules built mainly from hydrogen and carbon. Their first job is physical. They form part of the coating that slows water loss through the exoskeleton. Their mixture also provides information during close contact. A worker touching another ant with her antennae samples this chemical coating. The odor may contain clues about species, colony membership, worker caste, age, and reproductive condition. Colony odor is shaped by several sources. Genes influence which hydrocarbons an ant can produce. Food and nest material may alter the mixture. Frequent grooming and body contact help chemicals spread among nestmates, making colony members smell more similar over time. Recognition depends on comparison. A worker develops an internal expectation of the chemical patterns normally encountered inside her nest. When another ant's odor falls within an accepted range, the encounter may remain calm. A strongly unfamiliar mixture can lead to longer inspection, avoidance, alarm, or attack. The expected pattern can change as the colony ages, shifts diet, moves nest, or gains new workers. A queen's fertility signal works through the same general principle of chemical information. It reveals her reproductive condition to nearby workers and may reduce their tendency to develop active ovaries. The queen produces the signal as part of her biology. Workers respond according to inherited sensory systems and the colony's current condition. Every chemical message changes the likelihood of a response. Thousands of workers then encounter those messages, add new ones, remove material through cleaning, or allow older traces to fade. Signals guide the individual movements, while larger colony outcomes arise as those movements begin altering one another. A single trail may draw different responses from workers reaching it only seconds apart. One ant follows immediately because the chemical is strong beneath her antennae. A second worker pauses where the path divides and chooses the weaker branch. Another turns away after meeting a returning forager carrying no food. These small differences help a colony adjust rather than remain locked onto one choice. A rich food source creates positive feedback. Positive feedback means that an early response makes more of the same response likely. A worker finds a drop of sweet liquid, fills the expandable crop inside her body, and lays a chemical trail on her return. Other ants follow that route and reach the same reward. Successful workers reinforce the path during their own journeys home, increasing the number of ants using it. A colony can build a busy trail within minutes when the food is close, plentiful, and easy to collect. Several forces can weaken the pattern. The pheromone slowly evaporates. Crowding may delay workers along a narrow path. A predator can trigger alarm signals near the food. The supply may shrink until returning ants carry only small amounts. Workers that fail to find a reward add little or no new trail chemical. These effects create negative feedback, which reduces a response when conditions become less useful. Positive and negative feedback allow the colony to change direction without waiting for instructions. The same basic process helps ants make a much larger decision when their home is damaged. Temnothorax albipennis is a small ant found in rocky parts of Europe. A mature colony may contain only a few hundred workers and fit inside a thin crack beneath a flat stone. The entrance can be only a few millimeters wide. Inside, the cavity must provide enough floor space for adults and brood, while protecting them from rain, drying air, and sudden temperature changes. If the stone shifts or the roof opens, scout ants begin searching nearby. A scout enters a possible nest and spends time moving through it. She can assess the size of the cavity by walking across the floor and encountering its walls. Entrance width also matters because a narrow opening is easier to defend. Darkness provides information about how well the chamber is enclosed. Humidity, ceiling height, and the amount of exposed interior space can affect the safety of the brood. Different scouts may discover different cavities and begin favoring separate locations. No worker visits every available site. Each scout gathers only part of the information, then influences other ants through her behavior. A scout that accepts a possible home may return to the damaged nest and begin a tandem run. During a tandem run, one informed ant leads a single follower. The leader moves slowly enough for the follower to remain close, often touching her with the antennae. The pair may stop many times along the route. If contact is lost, the leader waits while the follower searches nearby. This slow journey allows the follower to learn landmarks and turns instead of being carried passively. After reaching the cavity, the follower inspects it for herself. She may later recruit another worker, creating a growing network of informed scouts. The colony can spend hours comparing sites in this way, when the old nest still offers some protection. Encounters gradually become more frequent at the better candidate. Scouts meet other scouts there, find workers already present, and return along increasingly familiar routes. Once activity rises above a response threshold, recruitment changes. This threshold is called a quorum. A quorum does not require every ant to cast a vote. It occurs when a worker encounters enough nestmates at a site within a certain period. That local experience signals that the colony is concentrating there. The scout then shifts from slow tandem running to rapid transport. She carries adult nestmates one at a time, curled beneath her body. Workers also begin moving eggs, larvae, and pupae between their mandibles. A relocation that began with scattered exploration becomes a steady flow toward one cavity. Danger changes how much evidence the colony requires. When the old nest is completely exposed, workers often commit more quickly. Speed becomes especially valuable because brood can dry out or be taken by predators. Under safer conditions, the colony can spend longer comparing entrance size, darkness, and internal space. This creates a trade-off between speed and accuracy. Fast choices reduce time spent in danger. Longer comparison improves the chance of selecting the better home. The final result resembles a colony making a judgment, although each worker still acts from nearby encounters and limited experience. Repeated recruitment strengthens one option until it crosses the collective threshold. Workers then turn that choice into movement, carrying the colony's living contents toward a new shelter. Such decisions depend on having enough ants ready to scout, transport brood, defend exposed routes, and repair the chosen cavity. That workforce must divide itself among many urgent jobs while conditions continue to change. Once a colony commits to a new home, its movement depends on workers responding to very different needs. Some carry larvae between their mandibles. Others guard the exposed route or begin clearing loose material from the new entrance. The workers performing these jobs are usually female. Most develop from fertilized eggs and never mate. Their ovaries remain inactive or only partly developed, allowing their bodies and energy to support the colony's shared work. Queens are reproductive females with bodies shaped for producing eggs. Males follow a shorter life path. They usually develop from unfertilized eggs, leave the nest during a mating period, and survive only long enough to reproduce. Their place inside the colony is brief compared with a worker that may spend months gathering food or caring for brood. The word caste describes a group within the colony that differs in body form, reproductive role, or typical behavior. Every ant species divides reproduction from most daily work, but the worker population can be organized in many ways. In some colonies, workers are nearly the same size. Other species produce a smooth range from small workers to much larger individuals. A few have clear physical groups that are easy to recognize. Many colonies have no dramatic soldier caste at all. Their defense is handled by ordinary workers using numbers, jaws, stings, or chemical signals. These differences begin while the ants are still larvae. A young female larva may have genes very similar to those of her sisters, yet she can develop into a worker, a large worker, or a queen. The result depends on how her growing body receives food and chemical signals. Rich nutrition can support longer growth and larger adult size. Temperature affects how quickly tissues develop. Hormones guide changes in organs, muscles, wings, and behavior. One important hormone is juvenile hormone. Despite its name, it remains active in adult insects as well as young ones. Its exact effect differs among species, but it can influence larval development, reproductive tissues, and the age at which a worker begins certain jobs. The colony therefore produces different adults by adjusting conditions during growth. A visible example appears in the large genus Phytolay, which contains more than 1,000 described species. Many Phytolay colonies produce small workers called minors and much larger workers called majors. A major can have a head several times wider than that of a minor. Large muscles fill much of this expanded head and power the mandibles. In seed-eating species, majors may crack hard seeds that smaller workers cannot easily open. They may also hold a narrow entrance against an intruder, or help cut apart a large piece of food. Their role changes among species and even among colonies facing different conditions. Minors usually perform most routine work. Their smaller bodies move easily through narrow tunnels and around crowded brood chambers. They forage, feed larvae, care for the queen, and maintain the nest. A major may also perform these activities when the need is strong enough. Physical form creates tendencies rather than an unbreakable set of rules. Age produces another useful pattern. In many ant species, recently emerged workers spend much of their time in protected areas near the brood. Their exoskeleton is still hardening, and their chemical profile continues developing. They clean eggs, feed larvae, and tend the queen. As workers grow older, they often shift toward tasks farther from the colony's center. Nest repair and waste removal may appear before regular outdoor foraging. This change in task tendency with age is called age polytheism. Polytheism simply means that members of the same species perform different kinds of work. Older workers often take greater risks outside because their remaining life expectancy is shorter. Still, age does not control every decision. A young worker may leave the nest early after many foragers are lost. An older forager can return to brood care when the colony suddenly produces a large number of larvae. Workers also differ in response thresholds. A response threshold is the strength a signal must reach before an individual reacts. One worker may begin moving brood after detecting a small temperature change. Another may require a stronger brood odor or more direct contact. Some workers respond quickly to loose soil around a damaged wall. Others are especially sensitive to hunger signals from nestmates. Because these thresholds vary, a small number of workers respond first. More individuals join as the need grows stronger. The colony gains a workforce that can expand or shrink around each problem without assigning jobs from a central point. This flexibility begins long before the worker reaches adulthood, inside the developing body of a larva. Yet the first larvae of a new colony have no workers to feed or protect them. Their survival often depends on one newly mated queen, sealed beneath the ground with stored sperm and the powerful flight muscles she will no longer need. Before any worker begins tending brood or carrying food, a new colony may exist inside the body of one winged female. She develops in a mature nest that has already gathered enough food to invest in reproduction. Her thorax is larger than a worker's because it contains powerful flight muscles. Males develop alongside her, usually with slimmer bodies, long wings, and a short adult life centered on mating. Winged queens and males are called elates. Their departure is often timed with weather that improves the chance of survival. Warm air, light wind, and moist soil after rain can bring thousands of elates into the sky within a narrow period. Releasing them together makes it easier for ants from different colonies to meet. It also overwhelms some predators because more insects appear than birds, spiders, and other hunters can capture at once. In many species, mating takes place during flight. A queen may mate with one male or several, depending on the species. Males usually die soon afterward. The queen lands away from her birth nest and begins searching for a suitable place to disappear underground. Once she has chosen a site, she removes her wings. She may grip each wing with her legs and pull until it breaks away at a prepared joint near the base. Small scars remain on the thorax for the rest of her life. Those marks reveal that she once flew, even many years after the colony has grown around her. The sperm collected during mating is stored inside an organ called the spermatheca. This small chamber connects with the reproductive tract. As an egg passes through, the queen can release a tiny amount of sperm to fertilize it. The stored cells may remain alive for years. Queens of some long-lived species continue producing fertilized eggs for more than a decade without mating again. Keeping sperm alive for so long requires carefully controlled conditions inside the spermatheca. The queen limits damaging chemical reactions and maintains an environment that protects the cells while using them in very small amounts. Her next steps depend on the founding method of her species. Many queens practice claustral founding. Claustral means enclosed. The queen digs a short tunnel, creates a small chamber, and seals the entrance behind her. A red imported fire ant queen may dig several centimeters into damp soil before closing herself inside. The chamber is dark, narrow, and scarcely larger than her body. She remains there while laying her first eggs. Food does not enter from outside. Stored fat provides part of the energy she needs. Her flight muscles provide another reserve. Those muscles once powered the wings, but they now begin breaking down. Proteins from the unused tissue are recycled and converted into nutrients that help support the queen and her developing brood. The first larvae receive food from her body. She may produce liquid secretions and lay some unfertilized eggs that serve as food. Her activity stays limited because every movement consumes energy that cannot easily be replaced. Other species use semi-claustral founding. These queens also begin inside small chambers, but they must leave periodically to hunt or gather food. Each trip creates risk. A queen outside the nest can be taken by a bird, trapped by a spider, injured by another ant, or dried by hot air. Even a sealed queen faces serious dangers. The chamber may flood after heavy rain. Dry soil can remove water from her body. Fungal growth may reach the eggs. Stored energy can run out before the first workers become active. A large share of founding queens die without producing a lasting colony. Those that succeed eventually see the first adults emerge. These early workers are often called nanitics. They are smaller than later workers because the queen raised them from a limited store of nutrients. Their bodies may be pale and soft at first. Within hours or days, the exoskeleton hardens and darkens. The nanitics open the sealed entrance and begin searching for food. They feed the queen, care for new larvae, remove waste, and enlarge the chamber. The queen's daily role changes as this first workforce takes control of outside activity. Her abdomen expands as egg production increases. She becomes the colony's main reproductive body, while workers regulate feeding, construction, defense, and brood care around her. The appearance of those first daughters ends the queen's complete isolation. A durable colony now depends on producing more workers before the earliest ones die, creating an unbroken flow from egg to larva, from pupa to adult. Inside the founding chamber, the queen's first eggs rest in a small, pale cluster. Each egg is only a few millimeters long, and the queen keeps the group close beneath her head and forelegs. She turns the eggs, cleans their surfaces, and gathers any that roll away. This care limits fungal growth and keeps the brood in a stable part of the chamber. Ants pass through 4 life stages: egg, larva, pupa, and adult. This pattern is called complete metamorphosis. The word metamorphosis means a major change in body form during development. A larva that hatches from an ant egg has no legs and little ability to move alone. Its body is soft, pale, and usually curved. The larval stage carries out most of the feeding and growth that will determine the adult's final size. Food may arrive as liquid from an adult's mouth or as small, solid pieces placed against the larva. In some species, larvae bend toward nearby food and feed themselves. In others, workers must hold the food directly against the mouth. As the colony grows, workers take over this care from the queen. They sort brood by age and move it through different parts of the nest. Eggs may remain in one cluster, while larger larvae occupy another chamber. Pupae may be placed in warmer or drier areas, where development continues more quickly. A few degrees of temperature difference can change how fast an immature ant grows. Humidity matters as well. Eggs and young larvae can dry out, while damp chambers may allow harmful fungi to spread. Workers sample these conditions with their antennae and move the brood when the surroundings change. After feeding and growing, a larva enters the pupal stage. Inside the pupa, larval tissues are reorganized into the body of an adult ant. Legs lengthen, antennae take shape, and the waist becomes distinct. Some species spin a silk cocoon around the pupa. The tan or brown bundles, sometimes mistaken for ant eggs, are usually these cocoons. Other lineages produce naked pupae whose developing limbs remain visible through a pale covering. The adult that emerges has reached its permanent size. At first, the exoskeleton is soft and light in color. Over the following hours, it hardens and usually becomes darker. The sex of that adult was largely decided when the egg was laid. Ants usually use a system called haplodiploidy. A fertilized egg normally develops into a diploid female, meaning it receives one set of chromosomes from the queen and another from stored sperm. An unfertilized egg normally becomes a haploid male, carrying only the queen's set of chromosomes. This difference separates males from females. The path from female larva to worker or queen is controlled by other influences. A diploid female can develop into either form. Nutrition is especially important. A larva receiving more food, or food with a different balance of nutrients, may grow longer and build the larger body of a future queen. Hormones guide the development of wings, ovaries, muscles, and other tissues. Temperature and colony condition can also affect the result. The exact combination differs among species, and some lineages use strong genetic influences alongside environmental ones. A young founding colony usually directs its limited food toward workers. The first adults, often called nanitics or minims, are small because the queen raised them from stored body reserves. Once they begin foraging, more energy enters the nest. That food supports larger larvae and a faster rate of egg production. Each added worker increases the colony's ability to collect food, care for brood, and enlarge the nest. The growing workforce also consumes energy, so resources remain limited. Adult workers require food. Larvae need protein and other nutrients for growth. The nest may need expansion, while food may be stored for difficult periods. Only after the colony reaches sufficient size can it invest heavily in males and new queens. Population rises through a repeating exchange between food and labor. Workers gather resources that support brood, and developing brood becomes the workers that gather future resources. Most of those adults will never encounter the original food source. Nourishment must move from the few ants returning from outside into the bodies of queens, larvae, and workers deeper within the nest. A returning forager may enter the nest carrying far more liquid than her own body needs. Inside her abdomen is an expandable pouch called the crop, The crop stores food before it reaches the deeper digestive organs. Because its contents can be shared, it is sometimes called the social stomach. A narrow valve separates the crop from the midgut. The midgut is the region where digestive enzymes break food apart, and nutrients begin passing into the ant's body. Liquid that remains in the crop can still be given to other members of the colony. Near the entrance, the swollen forager meets a worker that has spent the day underground. Their antennae touch briefly. Then the two ants place their mouthparts together. A small droplet moves from the crop of the returning ant into the receiving worker. This exchange is called trophallaxis. The word refers to the transfer of liquid food between members of a social group. Trophallaxis can occur between adult workers, from workers to queens, and from adults to developing larvae. The amount transferred during one exchange may be tiny. Repeated contact allows the original load to spread through many bodies. A worker that receives food near the entrance may walk deeper into the nest and share part of it with a brood carer. That brood carer can later feed a larva or pass a smaller amount to another worker. Within a short period, liquid collected by one forager can reach individuals that have never left the darkness of the nest. Distribution adjusts through local demand. A hungry ant is more likely to request food by touching and positioning herself near a donor. She may accept a larger amount before ending the exchange. A worker with a full crop takes less and may soon offer food to someone else. Foragers also respond to how quickly nestmates accept their loads. A returning ant that empties her crop rapidly may leave again for the same food source. Long delays can signal that the colony already has enough of that resource. Information about hunger therefore travels through the speed and size of the exchanges. No worker needs to count the colony's food supply. Thousands of brief encounters reveal where demand remains high. Trophylactic fluid contains more than sugar and water. Studies of the Florida carpenter ant, Camponotus floridanus, have found a complex mixture inside the exchanged liquid. It can contain digestive enzymes, proteins, chemical recognition cues, and compounds connected with growth. Researchers have also detected juvenile hormone in the fluid. Juvenile hormone helps regulate development and behavior in insects. Its effects depend on the species, the ant's age, and the amount present. When larvae receive trophylactic fluid, they may therefore receive nutrients alongside chemical signals that affect how quickly they grow. Some proteins in the liquid help digest food before it reaches the larva. Other compounds may influence the development of tissues or the timing of changes in adult workers. The exact mixture is not constant. Young workers can produce a different balance of compounds than older foragers. Colonies with many growing larvae may circulate fluid with a different chemical profile from colonies investing in winged queens and males. Diet also changes what enters the shared supply. A colony feeding heavily on nectar carries different nutrients from one receiving insect prey rich in protein. This moving mixture allows the colony's current condition to shape the bodies developing inside it. The comparison with circulation is useful within limits. Ants remain separate animals, each with her own digestive system and internal fluids. Trophallaxis creates a contact network that moves nourishment and biological information among those separate bodies. Its path depends on who meets whom. Workers near the entrance receive material first. Brood carers carry it toward larvae. Queens gain food through attendants that remain close to them. The structure of the nest helps determine how often these encounters occur. A narrow tunnel concentrates traffic. A large chamber allows workers to gather around brood. Temperature affects where larvae are placed, while waste and stale air must be kept away from growing bodies. Food movement is therefore tied to the physical arrangement of chambers, shafts, entrances, and passages. As the colony expands, the nest becomes part of the system that controls where workers meet and where its shared resources travel. A fed worker leaves the brood chamber and enters a narrow passage where another ant is cutting into the wall. The digging worker braces her legs against the tunnel floor and closes her mandibles around a small piece of packed soil. She pulls until the grain breaks free, turns, and carries it toward the entrance. Near the surface, she reaches a loose pile where other workers have already dropped similar fragments. The texture and odor of that pile affect where she releases her load. Her single action changes the surface that the following worker will encounter. This kind of coordination is called stigmergy. Stigmergy occurs when one animal changes its surroundings, and that change guides the actions of others. The workers can continue building without meeting or exchanging detailed instructions. A fresh soil pellet, an open edge, a chemical mark, or a partly completed tunnel can encourage another ant to continue the same task. Thousands of these local responses gradually produce a structure much larger than any worker can see. A simple nest may consist of one cavity beneath a stone. Larger colonies can build vertical shafts connected to chambers that widen sideways through the soil. A shaft acts as a passage between depths. Each chamber provides a broader space where workers can gather, brood can be placed, or food can be handled. These uses remain flexible. The same chamber may hold larvae during one part of the day and resting workers later, after temperature or humidity has changed. Researchers have studied nests of the North American ant formica pallidifulva by filling abandoned tunnels with casting material and carefully removing the surrounding soil. Many of these nests reached roughly 30 to 45 centimeters below the surface. Their main shafts descended through the ground, with flattened chambers opening at several levels. The chambers often became wider in older, larger colonies because a growing population required more usable floor space. Harvester ants can build much deeper systems. Species in the genus Pogonomyrmex may create branching nests that descend well beyond a person's knee and, in some cases, more than 2 meters into suitable soil. Deep chambers can protect stored seeds and brood from rapid changes at the surface. Architecture still varies with the local ground. Loose sand collapses differently from clay. Stones redirect tunnels, while roots may block one route or strengthen the roof of another. Heavy rain can flood lower passages, and dry weather can harden exposed soil. The colony responds by extending, sealing, widening, or abandoning parts of the nest. These changes arise from workers reacting to immediate conditions around their bodies. The nest also contains a series of climates. Sunlight may warm the upper soil during the day, while deeper chambers remain cooler. At night, the temperature difference can reverse. Moisture generally lasts longer below the surface, although saturated soil can become dangerous after rain. Oxygen enters from openings and moves through passages. Carbon dioxide produced by ants, larvae, and microbes must move outward. Some gases travel by diffusion, the slow spreading of molecules from areas of higher concentration toward areas of lower concentration. Wind passing over entrances can also move air, while temperature differences may create gentle currents. Certain mound-building species alter openings in ways that improve airflow. Every mound functions differently, and many rely mostly on ordinary physical forces acting through the nest's changing shape. Workers use these gradients directly. Brood may be carried upward when a warm chamber speeds development. Later, larvae can be moved deeper as the surface becomes too hot or dry. A blocked entrance may be reopened after carbon dioxide rises or traffic becomes crowded. Refuse can be carried toward distant chambers or outside piles, reducing contact with growing brood. The result is a structure that changes with weather, population, and the colony's current needs. Its walls shape where workers meet, how food spreads, and which parts of the brood receive warmth or moisture. Yet every chamber depends on openings to the world above. Water, prey, seeds, and building material must cross those entrances, sending workers into landscapes where the nest may vanish from sight after only a few steps. A worker leaving the nest may cross several meters of open ground before the entrance disappears behind stones, grass, or uneven soil. Some species solve this problem by following chemical trails laid by nestmates. Others rely more heavily on vision, especially when the ground is too hot, dry, or changeable for a scent trail to last. Ant navigation therefore differs with habitat. A forest worker may combine odor with familiar tree trunks and patches of light. An ant moving through leaf litter can use surface texture and close-range landmarks. Desert species often depend on the sky and a running estimate of the nest's position. One of the best-known examples is Cataglyphis fortis, a desert ant found on the salt flats of North Africa. These ants forage alone across pale ground where vegetation is scarce, and daytime surface temperatures can rise above 50 degrees Celsius. Dead insects provide food, but the worker must reach them quickly and return before the heat becomes dangerous. Her outward path may twist repeatedly as she searches. When she finds food, her journey home is often much straighter. This ability depends partly on path integration. Path integration means continually updating the direction and distance of a starting point while moving away from it. Each turn changes the estimated direction of the nest. Every stretch of walking adds to the estimated distance. The worker does not need to remember the exact shape of the entire outward route. She carries a changing internal estimate of where home lies from her present location. Direction comes partly from patterns of polarized light in the sky. Light from the sun scatters as it passes through Earth's atmosphere. This creates an arrangement that human eyes usually cannot see, with light waves pointing in different preferred directions across the sky. Specialized parts of an ant's compound eyes can detect these patterns. They remain useful when the sun itself is hidden behind a cloud or lies outside the ant's direct view. The sky, therefore, provides a broad compass. Distance requires a different source of information. Experiments with Cataglyphis ants showed that stride length contributes to their estimate. Researchers allowed workers to walk from a feeder back toward the nest. Some ants were fitted with tiny extensions that lengthened their legs, creating a stilt-like step. These workers passed the nest entrance and continued searching beyond it. Other ants had their legs shortened slightly. Their smaller steps caused them to begin searching before they reached home. The results showed that the ants were measuring distance partly through repeated movements of the legs. This process is often described as a step counter, although the real system probably includes information from muscles, joints, and the pattern of walking. Path integration works well over open ground, but small errors build during long journeys. A slight mistake in direction can become a large miss after several hundred body lengths. Ants reduce this error by adding other kinds of information. A worker may learn the dark outline of a shrub, the shape of a rock, or the open space between two grass stems. She can also detect familiar odors near the entrance and feel changes in the ground beneath her feet. These landmarks become especially important during the final part of the return. Young foragers prepare before making long trips. In several visually guided species, inexperienced ants perform learning walks around the nest. They leave the entrance in short loops and repeatedly turn their bodies back toward it. During these pauses, they view the surrounding scene from different positions. The nest itself may be a small hole with little visual detail, so the ant learns the wider panorama around it. Later, a familiar arrangement of trees, stones, and sky can guide her toward the correct area. Some ants also remember travel directions between important locations. A worker familiar with both a feeding site and the nest may take a new shortcut after being displaced, using stored directional information alongside visible landmarks. No single cue remains perfect under every condition. Clouds can change the available light. Wind may disturb odors, while moving leaves alter familiar views. The ant combines several incomplete systems, allowing one source to correct another. These short journeys connect the colony with food scattered across a large area. A few ant lineages eventually made that supply more predictable by carrying plant material underground and using it to cultivate a living crop. Along a forest trail, leafcutter ants move in a steady line beneath pieces of fresh green vegetation. Some fragments are wider than the ants carrying them. A worker grips the edge with her mandibles and holds it above her body like a small sail. The trail may continue for dozens of meters before reaching the colony entrance. The leaves are carried underground, yet the ants gain little nutrition by eating them directly. Plant cells contain cellulose, a tough material that most animals cannot digest without help. Leafcutters use the vegetation to feed a fungus. Two major leafcutter groups are Atta and Acromyrmex, found across warmer parts of the Americas. An Atta colony can contain several million workers and maintain long foraging trails through tropical forest or grassland. Large workers cut sections from leaves with repeated movements of their mandibles. Carriers transport the pieces home. Inside the nest, smaller workers take over. They clean the leaf surface and remove material that may carry unwanted microbes. The pieces are cut into smaller sections, crushed, and mixed with droplets from the ant's digestive system. This prepared plant material becomes the growing surface for a fungus in the genus Leucoagaricus. The fungus forms fine threads called hyphae. Together, many hyphae create the pale, sponge-like structure of the garden. Fungal enzymes break down parts of the plant material that the ants could not easily use on their own. The crop also produces swollen, nutrient-rich tips that can be eaten by the ants and fed to developing larvae. A healthy garden, therefore, depends on both partners. The ants supply fresh vegetation, maintain suitable moisture, and remove damaged material. They also carry pieces of the fungus when a colony moves or when a young queen begins a new nest. The fungus processes difficult plant tissue and turns it into food the colony can use. This partnership developed through coevolution. Coevolution occurs when two species shape each other's biology across many generations. Fungus farming began among the ant group known as the Atenes roughly 55 to 60 million years ago. These early farmers probably used dead leaves, fallen flowers, insect remains, or other decaying material as growing material. The highly specialized habit of cutting fresh vegetation appeared much later, perhaps around 15 to 20 million years ago. Human agriculture began only about 12,000 years ago. The ant system had already been changing through millions of generations before people planted the first domesticated crops. A leafcutter colony divides farm work partly by body size. Large workers can slice tough leaves and carry heavy fragments. Medium-sized ants process the material inside the nest. The smallest workers move among the fungal threads, tending narrow spaces that a large ant could damage simply by walking through them. Tiny gardeners remove foreign growth and inspect the crop with their antennae. Some also ride on leaf fragments during transport, where they may help defend carriers from small parasitic flies. The garden remains vulnerable to disease. A parasitic fungus called Escavopsis can grow through the crop and damage the cultivated fungus. Workers respond by grooming the garden and removing infected sections. Contaminated pieces may be carried to refuse chambers or outside waste piles, placed away from healthy growth. In some fungus-growing ants, bacteria living on the body produce substances that slow harmful microbes. These bacteria may appear as pale coatings on parts of the ant's exoskeleton. The colony's crop defense therefore combines cleaning behavior, chemical protection, and careful waste separation. Refuse workers can become strongly associated with waste areas and may have less contact with the healthy gardens. This separation lowers the chance that spores and decaying material will return to the brood chambers. A mature atta nest may contain many garden chambers arranged at different depths. Workers shift plant material and fungal pieces as temperature or moisture changes. The farm remains alive because millions of small adjustments continue each day. Leafcutters built their food system around one protected partner living beneath the soil. Other ants gain nourishment through relationships that remain above ground. Where insects produce sugary liquid, caterpillars exchange food for protection, and certain plants provide shelter for colonies living inside their stems. On a young plant stem, a worker ant pauses beside a cluster of aphids feeding from the soft tissue. Each aphid pushes a narrow mouthpart into the plant and draws out sap from a vessel called the phloem. Phloem carries dissolved sugars from the leaves toward growing roots, stems, flowers, and fruits. The sap contains far more sugar than an aphid can use, along with only a small amount of the nitrogen needed to build proteins. To obtain enough nitrogen, the insect processes a large volume of liquid and releases much of the extra sugar as a clear droplet called honeydew. An ant touches the aphid with her antennae. The contact can encourage the aphid to release a fresh droplet from the rear of its body. The ant drinks it into her crop and may carry the sugar-rich liquid back to the nest. For many colonies, honeydew provides a steady source of carbohydrates used to power walking, digging, defense, and other adult activity. The aphids can receive protection in return. A lady beetle larva approaching the group may be bitten, sprayed with acid, or driven from the stem. Ants may also remove the eggs of predators and disturb tiny parasitic wasps that try to lay eggs inside aphids. Some workers move aphids to young shoots where sap flows more easily. In colder conditions, certain ant species shelter aphid eggs or young aphids inside protected nest spaces before returning them to plants. This behavior is sometimes compared with herding. The comparison describes repeated guarding and movement, while the ants still respond through chemical signals, food rewards, and nearby conditions. The strength of the relationship varies. Some aphid groups receive only occasional visits. Other species depend heavily on ants and produce larger or more frequent honeydew droplets when attended. Protection can change the whole plant. Aphids remove sugar and other materials from living tissue. A large colony can slow the growth of a stem, curl young leaves, or weaken developing fruit. Honeydew that falls onto leaves may support dark fungi known as sooty molds, which coat the surface and reduce the light reaching the plant. By protecting aphids from predators, ants can allow their numbers to rise. The exchange benefits the insects while increasing the cost paid by the host plant. Other ant partnerships begin during the larval stage of butterflies. Many butterflies in the family, Lichenidae, produce caterpillars with glands that release sweet fluids. A caterpillar may also create vibrations or chemical odors that attract nearby ants. Workers gather around it, drink the secretion, and defend it from some predators and parasites. The large blue butterflies of the genus Fungaris use a more specialized path. Their young caterpillars first feed on plants, then drop to the ground and imitate the chemical scent of certain Myrmica ants. A worker carries the caterpillar into the nest, as though it were ant brood. Inside, some species receive food from workers. Others prey upon ant larvae. The caterpillar gains shelter by entering the colony's recognition system and altering how workers respond to it. Plants can also become long-term partners. In parts of Central America, swollen thorn acacias grow large hollow thorns that house colonies of Pseudomyrmex, ants. The plant produces nectar from glands near the leaf stalks. It also grows small nutrient-rich structures called Beltian bodies at the tips of its leaflets. Workers eat these food bodies and raise their brood inside the hollow thorns. The resident ants patrol the branches. They bite insects feeding on the leaves and may attack larger herbivores that brush against the tree. Workers also cut or damage vines and young plants growing too close to the acacia, reducing competition for light and space. The relationship is maintained because both partners gain useful resources. The plant receives defense. The ants receive shelter and a dependable food supply. Changing conditions can weaken that balance. An ant colony may take nectar while providing little protection. A plant may reduce its food rewards after damage or during drought. Natural selection can favor stronger cooperation, along with ways to take benefits while paying fewer costs. These living partnerships spread the colony's influence across stems, insects, and entire trees. An even quieter exchange begins when a worker finds a small seed with a soft, oily attachment and carries the future plant below the ground. A worker approaching the fallen seed finds a pale, fleshy structure attached to one end. This small reward is called an eleosome. Eleosomes contain fats, amino acids, or other nutrients that attract ants searching for food. The seed itself holds a plant embryo, protected by a harder outer coat. the ant grips the seed with her mandibles and begins carrying it toward the nest. For a creature only a few millimeters long, even a small seed can become a heavy load. The worker may drag it over leaves, lift it across roots, and stop several times before reaching the entrance. The distance is often less than a few meters, yet this movement can place the seed in a very different part of its surroundings. Seed dispersal by ants is called myrmekokori. The word comes from Greek roots referring to ants and movement. This partnership has evolved many separate times among flowering plants. Eliosomes appear in unrelated plant families, showing that the same basic exchange developed repeatedly under similar conditions. Inside the nest, workers gather around the seed and remove the soft attachment. They cut or scrape away the elaiosome and carry pieces toward larvae or other adults. The tougher seed coat often remains intact. Its embryo can survive the handling because the colony is mainly interested in the nutrient-rich tissue attached to the outside. Once the reward has been removed, the cleaned seed has less value to the ants. A worker may carry it into a refuse chamber. Another seed may remain in an unused passage or be taken back outside and dropped near the nest entrance. These final locations can improve the seed's chance of surviving. A seed left beneath the surface is hidden from some birds, rodents, and seed-eating insects. Soil can protect it from drying winds and sharp temperature changes. In fire-prone habitats, even shallow burial may place the seed below the hottest part of a passing flame. Nest refuse can also contain fragments of insects, dead workers, fungal material, and other nutrients that slowly enter the surrounding soil. A seed germinating there may begin life in a richer patch of ground. Movement away from the parent plant provides another benefit. Seeds falling directly beneath the parent compete with its roots and leaves. They may also gather in places where specialized predators and diseases are already common. An ant carrying the seed several meters away can reduce those pressures, even though the journey would seem short on a human map. Some woodland wildflowers depend strongly on this process. Trilliums, violets, bloodroot, and many other plants produce seeds with eliosomes. Across the world, approximately 11,000 plant species are thought to take part in some form of ant-assisted seed dispersal. The outcome depends heavily on which ant finds the seed. A large worker may carry it farther than a small species can. Some ants take seeds directly into deep nests, while others remove the elaiosome near the surface. Certain workers damage the seed coat or eat the embryo along with the reward. Others detach the elaiosome and leave the seed almost exactly where it fell. Those differences determine how much the plant gains from the exchange. Changes in the ant community can therefore alter the plants that appear in later years. When an invasive ant replaces a larger native species, seeds may travel shorter distances. A small invader may ignore heavier seeds completely, allowing plants with large seeds to decline near former dispersal routes. The effect may take several growing seasons to become visible. Fewer seedlings appear beyond the parent plants, and the pattern of vegetation slowly changes. The mechanism remains simple at the level of one worker. She responds to a food reward and carries it home. The plant gains movement as a consequence of that search for nutrition. Repeated across thousands of colonies and many generations, these short journeys influence where wildflowers grow, which seeds survive fire, and how plants spread through forests and grasslands. The mandibles performing this careful transport have taken many other forms during ant evolution. Some close around hard seeds with crushing force. Others seal nest entrances or release stored energy in movement so fast that the jaws become one of the most powerful tools available at miniature scale. A seed-crushing mandible can look very different from the jaws of an ant that hunts moving prey. Harvester ants often have broad, powerful mandibles suited to breaking tough seed coats. Leafcutters carry narrower cutting edges that act like small blades against plant tissue. Predatory ants may have curved tips that grip struggling insects. Trapjaw ants carry long mandibles held open in front of the head until a touch triggers their release. These differences reflect the problems each species faces. Food type influences jaw shape, while soil, prey, nest defense, and carrying behavior add other pressures. A worker that cuts leaves needs control during repeated slicing. An ant that cracks seeds benefits from a wide jaw supported by large head muscles. A fast-moving hunter gains more from long jaws that can close before prey escapes. Muscle alone cannot produce the fastest strikes. Muscles contract at a limited speed because their internal fibers require time to shorten. Trapjaw ants solve this limit by storing energy before the movement begins. The system is known as latch-mediated spring actuation. A spring-like structure inside the head is slowly loaded by contracting muscles. A latch keeps the mandibles open while energy builds. Sensitive trigger hairs project from the front of the head. When prey touches one of these hairs, the latch releases. Stored energy drives the jaws inward far faster than the muscles could move them directly. The ant Odontomachus bauri provides one of the best measured examples. This tropical species occurs across parts of Central America, South America, and the Caribbean. Its mandibles can reach speeds of approximately 64 meters per second. That is more than 200 kilometers per hour, during a movement lasting only a fraction of a millisecond. Acceleration can exceed 100,000 times the force of gravity. The total distance remains tiny because the jaws travel only a few millimeters. Within that space, the strike becomes one of the fastest movements measured in the animal world. The jaws may close around small prey before it has time to move away. A strike can also push an attacker back or create a sharp warning impact. Odontomachus ants use the mechanism for escape as well. When a worker snaps her mandibles against the ground, the force can launch her body upward. A downward strike may send her several centimeters into the air. A differently aimed impact can throw her backward from a threat. The landing is uncontrolled, yet the sudden movement can remove the ant from immediate danger. Other species rely more heavily on chemical defenses. Many ants retain a functional sting connected with venom glands in the gaster. The venom may cause pain, damage small prey, or discourage animals disturbing the nest. Its chemical mixture varies among species and can include proteins, peptides, and other active compounds. Formicine ants followed a different evolutionary path. This large group includes carpenter ants and wood ants. Their ancestors lost the functional sting while retaining glands that produce defensive chemicals. Many formicines release formic acid through an opening at the tip of the gaster. A worker may curl her abdomen forward and spray the liquid toward an attacker. Wood ants in the genus Formica can direct this spray across several centimeters, which is a considerable distance compared with their body length. Defense can also depend on body shape. Some ant colonies produce soldiers with heads much wider than those of ordinary workers. In certain turtle ants of the genus Cephalotes, a soldier's head forms a flat, rounded shield. The ant places this shield across the nest entrance, closing a hole in wood that may be only slightly wider than her body. Nestmates identify themselves through chemical contact, and the soldier moves aside for accepted workers. An intruder meets a living barrier supported by legs braced against the tunnel walls. This specialized body carries a cost. A broad-headed soldier may move less easily through narrow passages and perform fewer kinds of work than a smaller worker. Her value rises when the colony occupies a nest with entrances matching the shape of her head. Evolution has produced crushing jaws, stored energy strikes, chemical sprays, and bodies that become doors. Each defense operates through one worker's anatomy. Across a mass raid, hundreds of thousands of workers can combine those individual tools into a moving front spread across the forest floor. At dawn in a Central American forest, a dark stream of ants begins moving between fallen leaves. The line thickens as more workers arrive from behind. Near its leading edge, the narrow trail may spread into a broad front several meters across. These are army ants, a name used for several ant lineages that share a particular way of life. Their colonies contain enormous worker populations, conduct mass raids, and relocate frequently. Army ant queens are large reproductive females that remain wingless as adults. Their colonies also lack the permanent underground home used by many other ants. Similar traits developed in army ant groups in the Americas, Africa, and Asia. The New World genus Echiton provides some of the best-known examples. Echiton burchellii lives in tropical forests from southern Mexico through Central America and into South America. A mature colony can contain several 100,000 workers. Most remain hidden within the moving mass, guided by chemical trails and repeated contact with nearby nestmates. No worker can see the entire raid. Those near the front respond to prey, gaps, obstacles, and the movement of other ants within a few centimeters. A worker discovering prey releases recruitment chemicals and grips the target. Nearby ants arrive, increasing the local concentration of bodies and scent. The thicker trail draws still more workers from behind. Traffic can shift when one part of the front encounters more food than another. A blocked route redirects ants toward open ground. Crowding slows movement in one place and increases pressure along nearby paths. Thousands of local adjustments keep the raid changing shape as it advances. The animals taken by army ants are usually small enough to fit within this close-range hunting system. Workers capture insects and spiders. They enter the nests of other social insects and remove eggs, larvae, or pupae. Larger animals generally move away from the disturbance. Stories of army ants stripping every creature in their path greatly exaggerate their usual effect. The raid is still powerful at the scale of the forest floor. Small prey may be cut apart and pass backward through the traffic. Workers carry pieces toward the colony, while fresh ants continue moving forward. The raid also attracts other species. Antbirds follow nearby and catch insects fleeing through the leaf litter. Certain flies hover above the ants and attempt to reach prey before it is carried away. Beetles and mites have evolved close relationships with army ant colonies, sometimes living among the workers or traveling with them during relocation. Echiton burchellii follows a repeating cycle, connected with the development of its brood. During a more stationary period, often called the statuary phase, the colony remains in one location, while the queen lays eggs and larvae begin growing. As the larvae become active and require more food, the colony enters a nomadic phase. Raids occur more frequently, and the entire colony moves to a new resting place, often each night. Army ants in Africa can reach an even greater scale. Some driver ant colonies in the genus Dorylus contain several million workers. Their columns may extend over long distances through forest, grassland, or agricultural ground. Large workers defend the trail, while smaller workers move within the protected flow. Even at this size, coordination continues through scent, touch, and nearby movement. When an Eciton colony stops, its workers create a shelter called a bivouac. Thousands of ants link their legs and bodies together around the queen and developing brood. The outer workers form a shifting surface exposed to the surrounding air. Deeper layers create protected spaces where larvae and pupae can be held. Openings remain within the mass, allowing workers to pass through and helping air move around the brood. The bivouac may hang inside a hollow log, beneath a fallen trunk, or against sheltered vegetation. Its shape changes as workers enter, leave, or adjust their grip. The colony can dismantle the entire shelter when movement resumes. Bodies that hunted only hours earlier become the walls and support surrounding the next generation. This temporary nest reveals a wider ability within ant societies. Under changing conditions, workers can join themselves into structures that carry traffic, close gaps, float on floodwater, and reshape themselves while still alive. A small group of army ants reaches a break in the forest floor, where a branch, stone, or exposed root interrupts the trail. The first workers stop near the edge and grip the surface with their hooked claws. Other ants climb onto them, linking legs and bodies until the gap begins to close. Within a short time, the moving column has a living bridge. Workers carrying prey cross above the ants, holding the structure together. Traffic that once had to bend around an obstacle now follows a shorter route. The bridge forms through self-assembly. Self-assembly occurs when many individuals follow local rules and join themselves into a larger structure without receiving a complete plan. A worker feels the edge of a gap, encounters nestmates stretching across it, and grips nearby bodies. Ants already carrying food usually continue moving. Workers caught in the right location slow down and become part of the crossing. The bridge changes as traffic changes. A busy route encourages more workers to remain attached. When fewer ants approach, some members release their grip and rejoin the moving column. The structure can shift sideways as the main trail changes direction. Its length also adjusts to the width and angle of the gap. Experiments with army ants have shown that bridges often move toward positions that shorten the total route. A bridge placed closer to the inside of a bend saves more walking distance, but reaching that position may require more ants to form a longer structure. The colony therefore faces a physical trade-off. Every worker used as part of the bridge becomes temporarily unavailable for carrying food or joining the raid. A short bridge may use only a small number of ants, while saving many thousands of steps across repeated journeys. A longer bridge offers greater distance savings, but removes more workers from active traffic. Local responses allow the structure to settle near a useful balance. No ant calculates the number of bodies involved or measures the route with a map. Traffic passing over the bridge affects how long workers remain attached, and the changing flow gradually alters its shape. A different kind of living construction appears in the trees of Africa, Asia, and northern Australia. Weaver ants in the genus Acrophylla build nests by joining living leaves. Workers gather along two leaf edges and grip the surfaces with their mandibles. Additional ants form chains by holding one another's waists and legs. These chains can stretch across gaps wider than a single ant's reach. As the workers shorten the chain, the leaves are drawn together. Adult weaver ants cannot produce the silk needed to fasten them. Their larvae still possess working silk glands. A worker carries a larva gently between her mandibles and moves its mouth back and forth across the joined leaf edges. The larva releases a thin strand that sticks to the surface. Repeated passes create a pale woven seam. The larva becomes a living tool, while the adults control where the silk is placed. A finished nest may contain several connected leaves and house thousands of workers in the forest canopy. Water creates a larger challenge for ants living in flood-prone ground. Red imported fire ants, Solenopsis invicta, respond to rising water by leaving their tunnels and gathering at the surface. Workers interlock their legs and jaws until the colony spreads into a floating raft. Queens and developing brood are held within the mass, often toward protected central areas. The ants' outer surfaces resist water. This property is called hydrophobicity, meaning that water tends to bead and roll away rather than soak across the body. Tiny pockets of air remain trapped between linked workers. Together, the water-repellent surfaces and stored air help the raft stay afloat. The structure remains active throughout the flood. Workers move between upper and lower layers, changing which ants contact the water. A damaged edge can close as nearby ants attach themselves to the opening. The raft may bend around vegetation or reconnect after being partly divided. One colony can therefore change from an underground population into a floating sheet of living bodies. Bridges, leaf nests, and rafts all emerge from workers responding to contact, pressure, traffic, and nearby surfaces. Their stability depends on constant adjustment rather than permanent construction. Such close attachment requires reliable recognition. A worker joining a bridge must be accepted as part of the colony, while an unfamiliar ant approaching the same structure may be inspected, blocked, or attacked. At the edge of a nest entrance, two ants meet and pause with their antennae touching. Each worker samples the wax-like cuticular hydrocarbons coating the other ant's body. The scent is compared with a chemical pattern learned through daily contact with nestmates. Recognition works through degrees of similarity. A worker carrying a familiar mixture is usually allowed to pass. A strongly unfamiliar profile may cause a longer inspection, followed by avoidance, restraint, an alarm signal, or attack. The response can change with conditions. Hungry colonies may defend food more strongly. A nest filled with developing queens and males can become more sensitive to intrusion. Workers near a damaged entrance may react faster because alarm chemicals are already present. These encounters create boundaries around resources. Colonies compete for fallen insects, nesting cavities, seed patches, and groups of honeydew-producing aphids. Tree-dwelling ants may defend entire branches that provide food and shelter. Ground nesting species maintain routes between nest entrances and reliable feeding sites. The borders can shift from day to day. A colony that loses many workers may abandon part of its territory. Rain can erase scent marks and change which paths remain useful. A rich food source may draw workers from several colonies into the same small area, increasing the chance of conflict. Some territorial ants use repeated patrols to maintain their borders. Workers deposit chemical marks and respond quickly when they encounter unfamiliar ants. Fighting may involve biting, stinging, chemical sprays, or holding an intruder until more nestmates arrive. The intensity varies greatly among species. Some neighbors tolerate close contact, while others react to small differences in colony odor. Cooperation inside the nest also contains smaller conflicts. Workers and queens share much of their genetic interest because they are close relatives, though their best reproductive outcomes do not always match perfectly. A worker in many species can lay unfertilized eggs. Under the usual haplodiploid system, those eggs develop into males. Workers generally cannot produce daughters without mating, yet sons still allow them to pass on some of their genes. Other workers may remove these eggs. This behavior is called worker policing. A policing worker detects clues on an egg's surface, then eats it or carries it away. She may also direct aggression toward a worker with active ovaries. Policing can preserve the queen's reproduction and reduce conflict over male production. Some ants exploit an entire colony from the outside. A social parasitic queen enters the nest of another species or a closely related colony. She may carry chemicals that reduce the host worker's aggression or acquire the host odor after entering. In certain species, the invading queen kills the resident queen. The host workers then continue their normal care, feeding the intruder and raising the eggs she produces. As the older host workers die, the parasite's daughters gradually replace them. Other parasites remain permanently dependent on the host colony. These ants are called inquilines, meaning animals that live within the home of another species. Some have lost much of the body structure needed for ordinary work. Their lives center on reproduction inside a colony maintained by host workers. Brood care creates an especially useful weakness because workers respond strongly to young ants. Amazon ants in the genus Polyergus exploit this system through organized raids. A Polyergus colony depends heavily on workers captured from ants in the genus Formica. Raiders leave their nest in a column and enter a nearby Formica colony. They seize pupae and carry them home. The pupae contain nearly developed adults enclosed in cocoons or bare pupal coverings. When the captured ants emerge, they encounter the odors of the Polyergus nest during a sensitive early period. They learn that chemical environment as home. The new workers begin caring for brood, gathering food, and maintaining the nest that captured them. Polyergus workers are highly specialized for raiding. Their curved mandibles are effective during attacks, but poorly suited to many ordinary nest tasks. The stolen workforce supplies the labor their own bodies cannot easily perform. Recognition, brood care, and chemical learning usually hold a colony together. Social parasites succeed by entering those same systems at the right moment and changing how workers respond. A foreign ant still presents a body that can be touched and confronted. A fungal spore attached to that body may pass through the entrance unnoticed, placing the colony's constant contact against a threat far smaller than any worker can inspect directly. A worker returning from outside may carry fungal spores on her legs without showing any sign of infection. The particles can be smaller than grains of dust and may cling to joints, hairs, or the waxy surface of her exoskeleton. Inside the nest, close contact creates many opportunities for those spores to spread. Workers brush past one another in narrow tunnels. Food moves from mouth to mouth through trophallaxis, larvae are handled repeatedly by brood carers. Warmth and moisture protect developing ants, but those same conditions can also help fungi and bacteria grow. Most colony members are close relatives, so a pathogen able to infect one worker may also be well suited to infect her sisters. Ant societies reduce this danger through several overlapping defenses. The first begins with cleaning. A worker regularly draws her antennae through specialized comb-like structures on her front legs. Her legs pass between the mandibles, allowing dirt and small particles to be removed. Workers also clean one another in a behavior called allogrooming. The word means grooming another individual. A nestmate may scrape spores from places the exposed worker cannot easily reach, including the joints between body segments. Removing a spore early matters because many harmful fungi must first germinate on the ant's outer surface. A thin fungal tube then grows through the cuticle and enters the body. Grooming can interrupt that process before penetration occurs. Chemical defenses add another layer. Many ants possess a pair of metapleural glands near the rear of the mesosoma. These glands produce secretions that can slow the growth of some bacteria and fungi. A worker may spread the material across her body during grooming. Nestmates can also transfer small amounts through contact. Metapleural glands are common across ant lineages, although some groups have reduced or lost them. Their importance varies with species, habitat, and the microbes present. Other protective chemicals may come from venom glands or from microbes living on an ant's body. Fungus-growing ants provide a clear example. Certain bacteria associated with their exoskeleton produce compounds that help suppress organisms threatening the fungal garden. Nest material can contribute as well. Some ants collect tree resin and place it inside the colony, where its chemical compounds can reduce microbial growth. Sanitation keeps dangerous material away from vulnerable brood. Dead workers are often carried out of the main nest or placed in separate refuse chambers. Food remains and contaminated soil may follow different routes from healthy larvae. This separation lowers the number of contacts between waste and growing ants. Workers can respond before an infected individual dies. Research on the invasive garden ant Lasius neglectus has shown an especially direct defense involving disease pupae. A pupa infected with a lethal fungus may appear healthy from the outside during the early stage of infection. Workers can still detect chemical changes. They open the cocoon and remove its protective covering. The exposed pupa is then bitten and treated with antimicrobial substances. This process, called destructive disinfection, kills the infected individual before the fungus can complete its growth and release large numbers of contagious spores. The loss of one pupa reduces the danger faced by the rest of the colony. Protection also depends on the pattern of contact. Workers that spend time outside often meet nestmates near the entrance before reaching queens or young larvae. This arrangement can slow the movement of pathogens toward the colony's most valuable members. An exposed worker may receive more grooming or reduce her contact with others. Some infected ants leave the nest or remain in less crowded areas as their condition worsens. Colony traffic can shift around contaminated chambers, while brood is moved toward cleaner locations. These shared defenses are called social immunity. The term describes actions performed by colony members that reduce disease risk for the group. Small exposures can sometimes prepare an ant's individual immune system or encourage protective grooming among nestmates. the same contact network can spread a successful pathogen quickly when defenses fail. Fungi and other parasites continue evolving ways to remain hidden, resist antimicrobial chemicals, or produce spores before workers respond. The colony's close connections therefore create care and danger through the same pathways. Those pathways become even more powerful when people transport ants across oceans, placing entire colonies in regions where familiar competitors and natural enemies may be missing. A colony can cross an ocean without any worker knowing that a journey has taken place. A few queens may remain hidden inside a potted plant, a shipment of timber or soil packed around roots. Workers can shelter inside shipping containers, vehicles, produce crates, and other cargo that moves between ports. Most transported colonies fail. Some arrive in a climate that resembles their original home and find nesting spaces around gardens, roads, farms, or buildings. Species with several queens have an advantage during this process because a small transported group may still contain enough reproductive females to establish new nests. Flexible nesting also helps. An ant that can live beneath stones, inside wall spaces, or among plant roots has more options than a species requiring one narrow habitat. The Argentine ant, Linapithema humile, provides one of the clearest examples. It originated in South America, especially around the Parana River drainage and neighboring regions. Human trade carried it to other continents during the late 19th and early 20th centuries. Established populations now occur in places with mild, Mediterranean-style climates, including coastal California, southern Europe, South Africa, Australia, and parts of Japan. Argentine ants form systems containing many separate nests. This arrangement is called polydomy. Workers move between connected nesting sites while carrying food and brood. Many colonies also contain multiple queens, a condition known as polygyny. If one nest is disturbed, queens and workers may survive elsewhere within the network. These traits make control difficult because destroying a visible entrance removes only a small part of the population. Within the native South American range, neighboring colonies often remain chemically distinct and may fight at their borders. Some introduced populations behave differently. A limited number of transported founders carried only part of the species' original genetic diversity into new regions. Workers across broad areas may therefore develop similar chemical profiles. When ants from distant nests meet, their antennae sometimes detect enough similarity for peaceful contact. Resources can move among nests instead of being consumed in repeated territorial battles. A cooperative network of this scale is called a supercolony. One widely studied Argentine ant supercolony was reported across approximately 6,000 kilometers of southern Europe, extending along parts of the Mediterranean and Atlantic coasts. Workers collected from locations separated by enormous distances often showed little aggression when researchers brought them together. The network was not one continuous carpet of ants. Suitable habitats were separated by cities, dry ground, mountains, and other barriers. Chemical acceptance still connected many populations across a geographical range far beyond that of an ordinary colony. Reduced internal conflict leaves more workers available for outward expansion. Dense trails can dominate food sources quickly. Native ants may be displaced from nesting sites or prevented from reaching sugary liquids and insect prey. Changes then spread through other parts of the ecosystem. Native seed-dispersing ants may decline, leaving some plant seeds closer to their parent plants. Argentine ants also protect aphids and scale insects that produce honeydew. Heavy protection can increase the number of sap-feeding insects on crops or wild vegetation. Animals that depend on native ants may lose an important food source. In coastal California, for example, changes in ant communities have been linked with altered food conditions for insect-eating animals, including the threatened coastal horned lizard. Other invasive ants create different problems. Red imported fire ants build mound networks, defend food aggressively, and possess painful stings. Tawny crazy ants form dense populations with quick, irregular movement and can displace fire ants in parts of the southern United States. Each invasion depends on its own climate limits, nesting habits, reproductive system, and relationships with local species. Introduced ants may also escape some of the predators, competitors, parasites, or pathogens found in their native range. This temporary advantage is called enemy release. The protection is rarely complete. Disease can spread efficiently through connected nests, while drought or cold may reduce worker activity. Native competitors sometimes recover, and newly arriving parasites can weaken populations. Several invasive ant outbreaks have declined after years of dominance for reasons that remain difficult to separate. Climate, food supply, disease, and competition may contribute in different amounts. A supercolony, therefore, remains vulnerable despite its size. Its rapid expansion makes ant influence easy to observe because ecological changes can appear within years or decades. Native ants have produced slower versions of many of those effects for tens of millions of years, continually moving soil, carrying seeds, hunting small animals, and redistributing nutrients across nearly every land ecosystem. Across tropical forests, dry grasslands, city parks, farms, and deserts, ant colonies repeat the same small actions on an enormous scale. The global estimate of roughly 20 quadrillion ants becomes more meaningful when each individual is considered as a mover of soil, food, seeds, waste, and living prey. Their combined dry carbon biomass has been estimated at about 12 million metric tons. Dry carbon removes the water from living bodies and measures the carbon that remains, giving scientists a more consistent way to compare different organisms. Ant abundance is especially high in many tropical and forested regions, although numbers vary greatly with rainfall, temperature, vegetation, and soil. A patch of dry desert may support scattered colonies with large territories. A warm forest floor can hold many species within a few square meters, each using different foods or nesting layers. Every nest changes the ground around it. The biological mixing and movement of soil is called bioturbation. Earthworms perform it by swallowing and passing soil. Burrowing mammals create larger tunnels. Ants contribute through countless smaller excavations repeated over years. A worker may carry 1 grain of mineral soil upward from a chamber. Another brings a seed below the surface. Bits of leaves, insect remains, fungal material, and dead workers move through the same passages. This traffic transfers material both vertically and sideways. Tunnels create open spaces between soil particles, increasing porosity. Porosity describes the amount of empty space within soil. Greater porosity can allow air to enter and rainwater to soak downward more easily. The effect depends on soil type and nest structure. In loose sand, a tunnel may collapse quickly. Clay can hold a passage longer, but may drain more slowly. Around some nests, waste and food remains increased nitrogen and other nutrients. Microbes respond to the added organic material, changing how quickly it decomposes. Plant roots may grow more strongly near a nutrient-rich mound. Other ant species remove vegetation, collect seeds, or concentrate waste in ways that make nearby growth more difficult. Their ecological effect comes from the species involved and the conditions surrounding the nest. Food webs are altered just as widely. Predatory ants capture insect larvae, spiders, and other small invertebrates. Scavenging workers break down dead animals and carry fragments underground. Honeydew-collecting species protect aphids and scale insects. Leafcutters move fresh vegetation into fungal gardens, while seed-dispersing ants transport future plants across the ground. Ants also become food. Lizards snap them from trails. Birds search leaf litter for workers and winged reproductives. Some mammals tear open nests to reach adults, brood, or stored seeds. Specialized beetles, flies, mites, and caterpillars spend parts of their lives within ant colonies or around their raids. These connections mean that a change in ant abundance can spread beyond the insects themselves. Temperature has a strong influence because ants are ectotherms. An ectotherm relies mainly on outside heat to regulate body temperature. Warm conditions can increase walking speed and foraging until the surface becomes dangerously hot. Cold slows muscle activity and chemical reactions. Different species have different temperature limits, so warming does not affect every colony in the same direction. A heat-tolerant desert ant may gain longer periods of activity. A cool, adapted woodland species may lose suitable habitat. Drought changes soil moisture and plant growth. Fire removes cover while creating open, warmer ground. Urban development replaces deep soil with pavement, walls, lawns, and artificial heat. Agriculture alters vegetation and often moves invasive species through machinery, soil, or traded plants. As ant communities change, their usual work may change with them. Seed movement can decline when large native ants disappear. Soil mixing may increase around dense invasive colonies. Predation can shift toward different insect groups. Human engineers have begun studying the same local rules that allow colonies to function under changing conditions. Ant colony optimization is a computing method inspired by trail reinforcement. Possible routes are tested repeatedly, and successful paths gain stronger virtual signals. Poor routes gradually lose influence. The method has been used for scheduling, network design, and delivery planning. Swarm robots apply related ideas. Each machine follows simple nearby information, while the group searches, moves objects, or reorganizes after damage. Navigation researchers also study how ants combine sky patterns, step information, and landmarks with very small nervous systems. Living bridges and fire ant rafts have inspired work on self-assembling materials that can repair their shape. Engineers borrow selected principles from colonies while adapting them to machines and software. The full biological system remains more complex than any direct copy. Its planetary effect comes from ecological accumulation. A soil grain moved once changes little. 20 quadrillion ants repeating similar actions across generations can alter drainage plant growth, food webs, and the distribution of nutrients. The scale of the result now leads back toward the colony itself. These changes were produced by separate workers, yet the workers survived and acted through organized societies whose properties belonged to something larger than any one ant. Back at the edge of an ordinary path, the worker from the beginning continues toward the nest. She still has no picture of the chambers beneath her feet. The total number of workers, the amount of stored food, and the colony's future remain beyond what any single ant can know. Her decisions come from nearby information. A chemical trace alters her direction. Contact with a hungry nestmate affects how much food she shares. Warm soil, loose particles, recent danger, and the condition of her own body change what she does during the following minutes. Those limited responses become useful because they occur inside a connected society. Trail networks spread information about food and danger without creating a central map. Trophallaxis carries shared nourishment from a few foragers into the deeper nest. Eggs pass through larval and pupal stages, renewing the workforce as older ants die. The queen supplies most of the colony's eggs and preserves sperm gathered during a brief period of mating. Her role is reproductive rather than managerial. Workers regulate nearly everything around her through feeding, brood care, construction, defense, and foraging. The nest adds another layer of control. Chambers at different depths provide changing levels of warmth and moisture. Openings influence gas movement and traffic. Workers respond to those local conditions by shifting brood, widening passages, sealing damage, or carrying waste away. Disease defenses also belong partly to the group. Grooming removes spores before they enter the body. Antimicrobial chemicals slow microbial growth. Contact patterns and sanitation reduce the chance that infection will reach queens or young brood. Each defense is carried out by individuals, while its benefit appears across the colony. In 1911, the American entomologist William Morton Wheeler published an essay called The Ant Colony as an Organism. His work helped popularize the term superorganism for highly integrated insect societies. The idea compares a colony with a larger living system whose main functions are divided among specialized members. Queens concentrate reproduction. Workers gather energy and maintain the nest. Chemical signals connect separate bodies, while the brood replaces worn or lost parts of the workforce. The comparison becomes especially useful when a trait belongs most clearly to the colony. A bridge has no meaning as the achievement of one worker. The same is true of a fungus garden, a relocation decision, or a defensive response involving thousands of ants. Colonies that organize these tasks effectively may survive longer and produce more winged queens and males. Those reproductives carry the colony's inherited traits into new nests. Natural selection can therefore shape coordination at the level of the whole society. Close relatedness helps this process because sisters share many genes. A worker that raises related queens can still help shared genetic material reach future generations. Relatedness forms one part of the explanation. Food supply, nest structure, mating patterns, colony size, and ecological pressure also influence which social systems succeed. Ant colonies vary too widely for one simple model to describe them all. Some contain one queen, while others support many. workers in certain species rarely reproduce. Elsewhere, worker-laid male eggs are common enough to require policing. A colony may occupy one nest for years or spread across many connected sites. Internal conflict can remain low, or it may become a regular part of colony life. The superorganism idea therefore describes a level of integration rather than a single shared consciousness. Every worker has her own nervous system, sensory organs, and immediate responses. She can become separated from the colony for a time and continue moving on her own. Her long-term survival and biological role still depend heavily on the society that feeds, replaces, and coordinates its members. The same decentralized organization has produced fungal farms, desert navigation, living bridges, mass raids, disease control, soil movement, and invasive supercolonies. Across all these examples, the larger pattern arose from repeated contact among animals with limited information. The worker reaches the narrow opening and disappears below the surface. Above ground, she looked like one small insect crossing a short distance. Below ground, that movement joins a wider circulation. Food travels inward as chemical information spreads through nearby bodies. Brood develops in chambers that shift with heat and moisture. Thousands of separate lives continue feeding, repairing, sensing, and replacing one another, maintaining one continuous colony life.